lab there. So I will now hand over to the moderators, and we'll just give you the. I'll just give you the overview.
First of all, I would like to invite Professor Dr. Saeed Khan from UHS Karachi. He's PhD in molecular biology and postdoc from molecular biology from US, and Professor of Pathology at DOC International Medical College. Dr. Khan is working as a professor, and he had been serving as the head of molecular pathology section at DDRL UHS. He earned his PhD in microbiology from University of Karachi in collaboration with AKU, Karachi, Pakistan in 2010, followed by postdoctoral training in molecular virology from Mullins Molecular Retrovirology Laboratory, University of Washington, Seattle, USA. So, sir, please come to the stage and enlighten us on the topic of AMPP year in review. This is really the topic.
Before Said starts, I just want to add something that when Said started his molecular biology training, we had the pleasure of having him in our lab for a few years. And the only reason he left us was that we didn't have a PhD program at that time, and that's what he wanted to do. So he was a very, very good, person to have in the lab, and we are extremely happy, and I'm extremely happy that he's doing so well now.
That's when when I was at AKU, he became our PhD student. So we are proud of him. He has done really well.
Okay, then the बोलती बंद हो गई. Thank you very much, the chair of the session and my mentor and my teacher. What I learned at SIUT after my master, it was of course a great source for me to move forward, and a mentor or teacher like Dr. Rana, who taught us not only how to do molecular biology, but how to talk to people, how to behave, how to wear dress, and to talk to people as a moral support and as a teacher. So thank you very much, madam, and I'm so glad that we are having you here. And the story of Association of Molecular Pathology, it's not that very long. It's a very short and the newest organization. That's why I will be sharing the last 1 year that we had. A lot of events took place in this 1 year, and there are many things that we can share, but we have chosen a few of them that we can share with you at this forum. So thank you for giving me this opportunity that I'll be sharing these glimpses. We started last year being elected as a president, and my team with me, our vice president, Dr. Romina, Natasha is a general secretary, Dr. Arshi from LUMS as a treasurer, and the executive committee that we have at the moment, of course, our ex-president, Dr. Zara Hassan, she did a wonderful job to bring the society till now. Dr. Israr, Dr. Asghar, and Dr. Salma Batur, my direct teacher and mentor at SIUT. So this was a wonderful team that we were having, and running a society, establishing a society is not an easy task. It's a volunteer-based, beside working in the lab and the office, moving a society ahead is quite a tough job. The objective that we have achieved in 2003 was the registration of the society from the organization that we have almost covered and achieved. We'll be sharing the good news at the end. And the presence of the society, visibility of the society on ground, and different education areas that we arranged and conducted different workshops, seminar, and sensitization seminars and events. Now people know about the molecular pathology, and that's why you're here, that you know about the association. And we are even trying to make it more visible, active, and fruitful. crosstalk with the CPSP is being discussed in the general body meeting that the molecular pathology is not yet being recognized as part of the CPSP fellowship, but we have initiated this. We are getting tough tasks because it's not easy to get inside when there are already a lot of players and they give you very little space. But with the team that we are having and the support of our seniors, inshallah, we will make it happen. Bit and pieces we have moved in already, and Dr. Javeria is taking the lead on this. I will appreciate her efforts that she is being in contact with the CPSP, and we have covered a lot of ground already, but it's not an easy task, believe me, that we are getting tough situation over there, but inshallah we will get it done. These are some of the glimpses from the previous activities that we had in last year, starting from handing over from the last PAP conference. I appreciate the efforts of Dr. Zara Hassan. She did a wonderful job and provide us a platform from where we can start. We conducted different sessions at the CPSP. Dr. Israr, I appreciate his initiative. He started arranging workshops and seminar individually as well and at the CPSP forum. Then different other members, they conducted session over there. Then working with the Center of Biomedical Ethics and Culture Center, that was a great initiative, and I appreciate the efforts from Dr. Natasha. She took a lead, and other members are also on there who are contributing in this forum. We not only working from the office and the lab, rather we are supporting people outside the lab because molecular pathologists are directly linked with diagnosing different diseases, including bacterial, viral, and different other non-infectious diseases, and where molecular pathology is playing its role. We serve in COVID, we served in flood, we were in the front line in monkeypox, because molecular pathologists were providing solution to this. So different laboratories in Karachi and Lahore, KPK, Peshawar, the molecular pathology lab, they were providing solution for the measles that were at that time quite difficult. These are some of the glimpses that different labs were supporting the stakeholder in monkeypox. And now we have CCHF in Quetta, particularly in different parts of the Balochistan. Over there, again, the molecular biologists, they are taking the lead, and they are helping the health department and diagnosing these patients and finding the contact tracing. So these are the team from the Balochistan who are doing the molecular biology and finding the CCHF contacts make visible. So what we have vision for 2024 is to make it more visible, effective. We have different working groups, and if you have any suggestion, we will be more than welcome, happy to welcome you. Like hemo-oncology group, then different variant analysis, NGS group, microbial genetics, molecular oncology, and ethical framework for handling genomics data, and a fellowship with the CPSP. Again, I'll appreciate the efforts from Dr. Javeria. She is taking the lead on this, and we are in different training and technical session for students, researchers, to make this forum more effective, more useful, and a lot of people can contribute their service in this. So the good news that we have almost registered this association. We have received the first application group just day before yesterday, and hopefully by end of this year we will get a proper registration of the society. That was a long pending issue, I think, for last 8 years. That was not been possible, but in the last year, with the efforts of all our members, alhamdulillah, we have achieved this task. So registration of IMBP is now, inshallah, will be soon available. I will acknowledge all my team members, my Vice President, Dr. Romina, Dr. Arshi, Javeria, Dr. Isra, Dr. Natasha, and all our senior, Dr. Zara, Dr. Rana, Dr. Salma for all the support, and I believe in all the young microbiologists, molecular biologists who are working in this field. This is a very bright and useful field, and is provide innovation and solution for many of the diseases and issues that a lot of other colleagues they cannot find a solution for. Like we can help the microbiologists, we can help the histopathologists, we can help the hematologists, we can help finding out the solution with the chemical pathologists. So molecular pathology with the source of sequencing, NGS, and different molecular tools, we can bring new solution. We are just discussing that even we can help the microbiology. They are taking too long time for diagnosing the disease to declare that it's a sensitive or not, resistance or not. So we can just using these molecular tools, innovative tools, can use these and can help the patient, the community at large, and we can also work on the prediction of different diseases. Like we witnessed the pandemic. So when the next pandemic come, when the COVID, we were just observing this data on daily basis and we providing the data to the authorities, the policy makers, and they were taking decision on this. So with the public health authorities that we are get connected, we can predict different diseases in our own locality, which can be helpful for us and the countries in future as well. So thank you very much for your time. If you have any question, I will be more than happy. Soon we will be starting the membership that is pending because of the after registration we can open the account, and we will be then opening everyone can become member. We can circulate that news, but that will happen after the registration. We are waiting for the official registration. Okay, no question.
So I just asked one question. Yes, I think relevant. So definitely we are doing a lot. So that was really nice that you presented the progress of molecular pathology. We are now ahead of the last year where we were. So the one thing which I suggested, and I just want to know the update, that obviously now in modern era, everybody visit a website, which is easy to know what's going on, what the next plan we have, or maybe some small queries if they have to ask us or suggest any event or maybe some activity on their location. So I remember I give this suggestion to Dr. Natasha a few weeks back, so I just want to know the update on this.
We have already worked on it. It's a very excellent idea. This compulsory that we need to have a website. For the website, we were just waiting for this registration as well, because until we have proper legal registration, after that we can have proper documentation of everything. So everything is ready. So
meanwhile, any other route? So there are a lot of people. I can see the excitement. So I wish we should provide them some sort of connectivity with us.
Okay. In the meanwhile, I think okay, we can provide the Facebook page that works for a while. Until we get this registration, then we will launch proper organized website. But your idea is, of course.
Actually, that's not my idea. This was actually wonderful work done by Dr. Meena Kazi and Dr. Natasha. All our workshops were very successful. People are now willing to be connected with us. They are approaching us, and we want some central place rather than.
We will start the new year with the new website.
So that I think we got good news for your queries.
Okay, can I just add one thing, that you don't have to wait for the registration of the society to develop a website. So maybe all what you really need is somebody who is willing to do it, because you need a volunteer, right? Because I don't know how much money we can pay at this time, right? So you need someone who can take this responsibility, who has some experience in developing a website, and maybe Dr. Saeed and Dr. Natasha, you guys can make a committee or something, and somebody give someone this responsibility. Because अगर आप किसी को देंगे नहीं तो बनेगी नहीं. हमने जो इम्यूनोलॉजी की सोसाइटी है उसकी भी जो अपनी वेबसाइट है वो हमारे एक मेंबर बनाते हैं, ठीक है. और उसमें फिर ये होता है कि आपको कुछ अगर ईयरली पेमेंट कहीं करना है, कुछ करना है, you can do it, but you don't have to be registered to do that.
डायरेक्टली अगर हम जरूरत नहीं थी कि हमारा अकाउंट जब तक खुलेगा तब रजिस्ट्रेशन भी, तो जब तक फिर से हम ट्रांजेक्शन करेंगे तो we can do it a volunteer basis at the moment.
Yeah,
And then
I think generally what we should have is a group working on communications, and I think communications, the team that can develop, Instagram posts, they can develop a Facebook, they can develop a website. So I think we need the working groups that we had out there. We probably need the first one we need is a communications group. Okay, so we can put that together. If anybody's willing to volunteer to actually be part of that, please just reach out to us, and we'd really like somebody to take the lead on this.
Assalamualaikum, I'm Dr. Humayun, Dhanzi University से assistant professor. मैं जो suggest कर सकता हूँ ये था कि जिस तरह बाकी यूनिवर्सिटीज में होता है कि आपके एक सेक्रेटेरिएट होते हैं जहाँ ह्यूमन रिसोर्स होता है जो डेडिकेटेड होता है. Doesn't need to be a senior official, but ये है कि आपके पास secretarial staff होता है. Number one, वो websites को manage कर सकता है, आपके Facebook pages को manage कर सकता है. Once that registration things comes in, तो वो उसको भी manage करता है. Queries वहाँ से regulate हो सकती हैं. But sometimes it does not involve much of the cost because PSA जो पाकिस्तान सोसाइटी ऑफ हेमेटोलॉजी है उसका जो सेक्रेटेरिएट है हमारे हॉस्पिटल में काफी लंबे टाइम से भी बेस्ड है. तो हम देखते हैं all it needs is 2 people who are just paid doing a paid job. वो शायद इतना ज़्यादा कॉस्ट भी नहीं करता लेकिन ये है कि उन्हें पता है कि this is their job. वो हम जो वॉलंटरीली बेस पे हमेशा इसको एडवोकेट तो करते हैं बट जब तक किसी को ईयर मार्क नहीं होता किसी बंदे की डेडिकेटेड जॉब नहीं होती तो फिर वो आपको उसमें गैप्स नजर आते हैं. That's one of the
Sir, आपका बहुत अच्छा idea. We are toward that, but again I will say that until we have this bank account, we cannot pay someone officially. So at the moment we are doing, everyone is doing a volunteer basis. For future, of course, we need to have a secretariat that we have in our vision, and we will hire people for the office who can do this work, like other society. I agree with you.
If I dare to propose one thing, कि इतने बड़ी-बड़ी ऑर्गेनाइजेशन है जिनसे हम लोग बिलोंग करते हैं, उनके अंदर आप सिर्फ नॉट सैलरी, आप ऑनरेरियम के नाम पे एक छोटे बच्चे को जो ग्रेजुएट है जो आईटी में थोड़ा एक्सपर्ट है उसको टेम्परेरीली अभी एंगेज कर ले सो दैट कि आपकी लीगल साइड कवर होते हैं फिर वो उसके बाद सैलरी स्ट्रक्चर बना लेते हैं. और इमीडिएटली शायद हम एक व्हाट्सएप ग्रुप से स्टार्ट कर सकते हैं. यहीं पे आप एक रजिस्ट्रेशन पेज रखें, लोग अपना मोबाइल नंबर वहां डॉक्यूमेंट करें सो दैट हम इसके बाद इस कॉन्फ्रेंस के बाद भूलना है कनेक्टेड रहे, हमें आपसे कनेक्शन मिले, आपसे हमारे साथ रब्ता रहे. मेरा पहला विजिट है और I would love to be a member as soon as this registration comes in.
We already have a WhatsApp group. You can be part of that immediately after this session. You can share the details with us. You can be part of. So on social media, WhatsApp people are already members, but officially we are waiting for the
Hospital
process.
में Natasha से share हुए. Thank you.
Okay,
sir.
thank you very much. Thank you, Dr. Said.
Thank you.
I think we need to move to
Thank you.
Thank you very much. Our next talk is on the topic of next generation sequencing, NGS expanding its role in molecular pathology. It will be presented by Dr. Samuel Paula. We have honor of his presence. He works at Illumina, UAE. Dr. Paula completed his PhD at the University of Toronto, Faculty of Medicine in the Department of Laboratory Medicine and Pathology. And he also completed his master's degree at the University of Waterloo School of Pharmacy with a focus on gene therapy and nanotechnology. Dr. Paul has worked extensively in the genomics emerging markers, and he is very passionate about making personalized medicine a cornerstone of healthcare system in the region. His areas of expertise include oncology, companion diagnostics, reproductive genetic health, and rare genetic diseases. So I welcome Dr. Paul to present his talk.
Thank you for the introduction. Hello everyone, welcome. Thank you for joining us here today. My name is Samuel Kalat. I'm from the Illumina team here, and we're honored to be in your presence. Just going to quickly start with a short clip that I think really nicely captures our mission at Illumina, and then we'll get started.
Powerful things, genes. They tell every living thing precisely what to be, what to look like, how to grow, how to flower, fertilize, reproduce, everything. But the Illumina knows that because genes determine who we are, they've also got the power to tell us what to do. By seeing genomics in a brighter light, we harness their power to build a stronger world, to keep one step ahead of a virus so it doesn't step on us, diagnose illnesses before they get the chance to make us ill, even nourish the entire world without starving it of resources. Because as a world leader in genomics, we know that the most powerful tools to improve the lives are the very things we're made of. Illumina.
So a nice video to capture the breadth of applications in which we can apply genomics today. And as many of you know, our mission in Illumina is to improve human health by unlocking the power of the genome. And today, Illumina is taking an active role to be closer to you. And as you can see, this is a map of our presence worldwide. And most recent addition to our offices in the region is the Dubai Solution Center, which we inaugurated last year. It's a complete training center and an engineering workshop that offers our partners and customers opportunity to learn our different applications and workflows, in addition to give the engineers in our network a chance to have very high level of training to support you and enable you. So as we saw in the video, the breadth of genomics applications is very wide. And this slide, I think, captures that nicely. Genetic disease, oncology, reproductive health, and research. So today, I chose to focus into two of these areas. The first one is genetic disease, because I think it's quite relevant in Pakistan and in the Middle East as well, in addition to oncology. So I would like to give you our vision at Illumina for genetic disease diagnosis. I know that there are some technical and sometimes economical constraints to that, but we would like to give you our dream, so to speak, where we want to see sequencing going in this region and where we hope it will be in a few years. So this story is about two individuals who were affected by genetic disease. And you can see here their journey was quite long for diagnosis, 6 years. So Carson and Chase, Carson is the older boy, and Chase is his younger brother. At about 1 year old, the parents noticed there was some movement issues with the child. At 2 years, the child was diagnosed with cerebral palsy. When Chase was born with similar symptoms, the original diagnosis of cerebral palsy was called into question because it's quite unlikely to have two siblings back to back have this diagnosis. At age of 4, whole exome sequencing came back negative, and finally, at the end of the 5th year with whole genome sequencing, they were able to identify a rare mutation in the MEPAN syndrome that was enabled this child to have the proper treatment and the proper diagnosis. So when we think about rare disease, it may be rare on case-by-case basis. However, collectively, it's not so rare. There are 6,000 characterized rare diseases to date. They affect in the US about 1 in 600 individuals, and 80% of rare diseases have a genetic component. It takes about 8 physicians on average to treat a case, 5 to 7 years for the duration of the process, between, of course, targeted panels that come back negative, whole exome testing that comes back negative, and finally whole genome, sometimes gene expression analysis to get to a diagnosis. The cost of that is quite massive, and I want you to keep that in mind when we talk about whole genome sequencing. I think when we say whole genome sequencing, people immediately think that it's expensive, but when you look at the average cost of treatment for a patient, it's nearly $20,000. It far exceeds the cost of a whole genome sequencing. So we hope to give patients the access. We want all the patients to have whole genome sequencing as a first-line test in diagnosis. It's not a dream anymore. We have already sites in the Middle East that are offering whole genome sequencing as a first-line test. We also want it to be a standard of care and expand it beyond whole genome sequencing to look at other omics, proteomics, transcriptomics, and other omics that complement the treatment and diagnostic plan. Whole genome sequencing is not just something that you have when you have a rare disease. So this chart here shows the different contexts in which someone could be eligible to have
whole genome sequencing, whether it's a newborn in the NICU, whether it's a child with symptoms at early ages, also adults. We have many adult onset diseases that require whole genome sequencing, and for identification purpose, also postpartum whole genome sequencing can be relevant. At Illumina, we want everyone to have access to whole genome sequencing because, as you can see here, with respect to the different variants that you can identify, whole genome sequencing gives you the biggest insight, even when you compare it to whole exome sequencing here. So it does a really good job if you have a targeted panel or whole exome sequencing. If you're working with single nucleotide variants, insertions, deletions, exome does a good job, but it does struggle a little bit when you're working with copy number variants. In addition to repeat expansions, as we know, there are many mutations in this domain that are missed with exomes, and finally, paralogs is something that you need whole genome sequencing to capture. Through many breakthroughs in R&D, technology and development, we have managed to bring down the cost of whole genome sequencing. It's still expensive, we understand that. So the first one was a little bit expensive, it was only $3 billion, but we've managed to bring that down to about $200. And we hope that as we continue to implement this technology, that the cost will be more and more affordable. You've seen this, I'm sure, in some of our marketing campaigns. This is the NovaSeq X Plus. This is the latest innovation from Illumina. It is able to sequence 162 whole genome sequences in a single run and do the analysis on board in an automatic fashion. So just wanted to share this really exciting developments happening in this field, and the technology is enabling us to do more with less cost. So just to give you an idea about what the X Plus can do, it can essentially double the throughput of our previously highest throughput instrument, which is the 6000, and it can do that in half of the time. And as we're seeing with many sites, the cost is also being cut in half. So today in Pakistan, we discuss a lot of targeted panels, small panels to get labs started, but very soon I would love to see the first site in Pakistan doing this locally, whole exome sequencing and whole genome sequencing on an instrument. So just to give you a quick picture of how do we implement as Illumina, how do we set up a site with whole genome, whole exome sequencing, or even targeted panels for rare disease. So the first step is to set up a sample management system. So our portfolio includes a product called Clarity Lens that allows the clinician and the clinical team to keep track of the samples, manage the workflow, prepare master mixes, record QC data, and control the roles and permissions of different lab members to ensure that samples are monitored and controlled from the beginning of the workflow all the way until the end. It is fully integrated with Illumina sequencers and software products, and it allows end-to-end sample traceability. So in terms of library prep, in addition to the Illumina DNA PCR-free from blood or saliva, which does whole genome sequencing, we have a similar workflow that allows exome sequencing for smaller focuses, and we also have an enrichment-based assay that can do focused panels using hybrid capture chemistries. So these are the latest two sequencers that we are positioning in the market to allow rare disease diagnosis. Both of these, the NextSeq 2000 is ideal for exome sequencing, and the NextSeq X series is ideal for both exome and whole genome sequencing. Both of these sequencers have onboard secondary analysis. So once the run is completed, you can go and you can download your VCF. And if you have our tertiary analysis tools, the system can be set up to push the VCF into tertiary analysis and prepare the PDFs automatically. So that's from a sequencing point of view. For smaller panels, which are important, sometimes after some time, scientists in a specific country can identify a subset of genes that they just want to focus on in a smaller panel. So that can go into our MySeq instruments and can do a really nice job there. So storage of data is a big challenge, and we have the Illumina Connected Analytics, which is a virtual space that allows you to store your data at an annual premium. And it allows you to access our Dragon in addition to customize your own informatics pipelines. Data analysis on our famous Dragon, which is a hardware/software secondary analysis tool that exists in many different forms. It can be part of your instrument, it can be on-site server, it can be virtual as well. And these are push-button secondary analysis workflows that allow you to go from the FASTQ to the VCF forms of the files in a highly accurate, reliable, and standardized way. Just to give you a picture here, so these are the different applications that our Dragon software enables. So it goes all the way from the early processing of the reads, trimming, conversion, PCL conversion, and compression to ensure that the data footprint of your workflow is minimized. It covers genetic disease, all of these variants that we discussed as part of a rare disease portfolio, and it also supports oncology and other research focuses. Finally, we are working to enable our customers with Imaging, which is our tertiary analysis platform, which allows you to do the interpretation and the issuing of the report. Just wanted to add quickly that short read sequencing has been able to do amazing things with regards to small variants, but we do know that there are large structural variants that are spanning beyond the short read realm. And some of you may have seen this. This is a comparison that was done as part of the Precision FDA competition, the Truth Challenge competition, where you can see that our data in orange here is comparable in accuracy to some of the on-market long-read chemistries. And we're able to achieve this with short read only, without accessing or without needing to go to long-read chemistry. We're able to do equivalent accuracy in long-read without going to long-read. However, as you know, last year, Illumina launched their long-read solution, which is compatible on our NovaSeq, and it's able to do some of the variants that are harder to access with the short-read chemistries. So you can see that you can access all of these variants with our short-read and Dragon platform, but with Illumina Complete Read, you can do phased sequencing up to 200 kb to resolve haplotypes. You can improve the resolutions in the short repeats and expanded structural variants to access those variants that you're unable to access with the short-read chemistry. This is just a quick comparison to show you how our long-read chemistry is compared to some of the on-market long-reads. So as you can see, this is a challenging medically relevant gene called SEMG1. And as you can see, the secondary analysis shows that we can capture those challenging regions just as good as on-market solutions. I want to talk a bit now, switch gears a little bit and talk about oncology testing, because I think that's a very important part for when we talk about pathology. All of the experts here know, understand the challenge of the patient's journey across the different stages of diagnosis, therapies, adjustment of the therapeutic plans, and how the sample starts from the histology part to clinical staging, to staining, identifying MDX markers, therapy, and sometimes, if the patient has access, monitoring of the disease as it progresses. In Illumina, we have a spectrum of solutions that cover very small panels. So some of you know we recently released the pillar panels, which are small solid tumor and myeloid panels that enable you to profile samples in small batches and looking at small targeted regions. So these can be applicable on the small sequencers that you see here. Some of you know those, the iSeq, the MiniSeq, and the MySeq. But we also have the very large comprehensive genomic profiling panels, which are compatible on the NextSeq and the NovaSeq. In addition to throughput, we can also slice this analysis by looking at germline versus somatic. So our TrueSight hereditary cancer panel, for example, is able to look at about 112 genes in one run. So instead of just doing BRCA1 and BRCA2, we're encouraging our customers to consider adding additional content by instead of just looking at BRCA1 and BRCA2, you can look at 112 genes and additional SNPs. But somatic variant detection, we understand, is a critical part of therapy, biomarker research, and diagnosis. And we finally have the methylation epic array that helps clinicians classify tumors and understand the source of the tumor and the tissue of origin. Again, today we're talking about vision, right? We're talking about where we want to see Pakistan in a couple of years. So this is just a timeline of the number of biomarkers or the number of therapies, approved therapies in the US from 2003 to 2021. Not for all cancers. This is for one type of cancer only. Look how many therapies are approved for non-small cell lung carcinoma, huge number of drugs. And the point of this slide is to show you that every year there are additional biomarkers that are discovered, additional therapies, and it's important to ensure that clinicians and pathologists have access to the latest and most comprehensive content in order to maximize the clinical utility of your workflow. Beyond non-small cell lung carcinoma, the number of biomarkers that are discovered every year is growing. So take a look at this slide here. These are just small number of biomarkers for different solid tumors. Each one of those is typically done in a smaller panel. And there are other pan-cancer biomarkers like INTERC, TMB, and MSI that do not fit in a small panel because of their large size. So when you put this in the context of the multi-workflows that you have in a typical lab, you can see that a typical lab would have a PCR team, a FISH team, an RT-PCR team, and a PCR team for other biomarkers. As you can see, these consume a lot of your time, a lot of your precious FFPE slices, which you may not have, and it complicates the overall picture. So what we are hoping to do is we're hoping to consolidate with time all of those tests into just one workflow where we use comprehensive genomic profiling to look at roughly 500 genes, to look at the pan-cancer genes like TMB and MSI, and consolidate all of those testing in one workflow. So in one study, just to give you an example of the power of comprehensive genomic profiling, and it's something that we hope that we will get there one day in this region. So one study looked at the clinical utility of using large panels like comprehensive genomic profiling for different studies of different sizes, so 300 patients up to 10,000 patients. And you can see the detection of actionable alterations was very impressive across the different studies, which shows you that the more you increase your content, the more chances you have of diagnosis. Very quickly here, just the large number of tests. Large panels are becoming increasingly implemented as a recommendation by European societies and oncology bodies in the US. So the message here that we know that a lot of the scientists here in the room and the clinicians are working with small panels, but we do hope that we will head into a direction to implement large panels as a consolidated workflow. Just wanted to check time. Does anybody know how much time? How much time? Five more minutes? We're at time? 2 more minutes, okay. 2 more minutes. I'll get through it in 2 more minutes. So I just wanted to show you our collaboration with Pillar Biosciences. So we've inserted the Pillar Biosciences portfolio in our workflow here to be compatible with the MySeq and even smaller, in addition to the NextSeq. So these panels include the multi-cancer panel that cover a large number of solid tumors. They also include the myeloid panel that looks at MPN, AML, CML, and MDS. And we have an essential MPN panel that looks at the JAK2, CALR, and NPL biomarkers. We have a BRCA1 and BRCA2 panel that also looks at CNV, which is quite nice to incorporate using FFPE in addition to blood and saliva samples. The last section here is very quickly is the tertiary analysis portfolio that we have in Illumina. We're excited to present this to you as a tertiary analysis tool that allows you to complete your tertiary analysis following the Dragon secondary analysis tool. It gives you access to a massive knowledge database to look at biomarkers, drug labels, and clinical trials. And it uses the Jackson Laboratory database, which has 44,000 variants, hundreds of guidelines and drug classes. It's compatible with solid tumor tissue, liquid biopsy, and hematological malignancies. And this is an example report just to show you what you're able to do. It gives you the mutation, the matched therapy, all the active clinical trials on the region, and additional genomic biomarker trials. Thank you so much for listening, and I'm happy to take up any questions.
Thank you, Dr. Sami. It's a wonderful talk. So I will request audience to hold their questions. First, we have to proceed further.
Sure.
And then we can discuss. We can take all the questions together.
Absolutely.
Thank you. So our next speaker is Catherine from she's from Oxford and Technology, UK. Ms. Warren manages commercial operations, direct sales, and channelize partners across the Gulf and Central Asia with Oxford Nanopore Technology. She graduated in 2017 with a 2.1 degree in biological sciences from Oxford University, and in 2019 with a pre-med post-bac certificate from Columbia University. So, please welcome Ms. Catherine Warren.
Hello, everyone.
Hello.
I'm pulling this up, but it's nice to meet you all. You might not even need this to hear me because I'm quite loud. I realize in general, which is when I brought the water. While they pull this up, I know we're running a little bit behind. It's a pleasure to meet you all. It's a pleasure to be here. It's my first trip to. It is my first trip to Pakistan, so I'm actually really honored to be here. And I'm sorting this out. Do my little bit of intro in the meantime. So yeah, keep you warm. They give you a little bit of my background, which is nice and unexpected. I'm part of Oxford Nanopore Technologies. I am on the commercial team based out of Dubai, although again from the accent you can tell I'm from the US. I'm from New York originally, and I did study at Oxford, so this is actually part of my alma mater, which is really cool. As it started about 16 years ago. I now manage all of Central Asia, parts of the Gulf, Middle East, and the region around here. So I'm going to walk you through a bit of the more clinical side of our technology today. We are obviously useful among many technologies for livestock, agriculture, agri-tech, and also things like metagenomics in the realm. So this is just a small bit of what we do, and if you guys have more questions about anything, please come and find me. We'll get those later on, or get my email later. So, Nanopore Technologies. Anything, anyone, anywhere. Nanopore has been used not only in now clinical services, not only in research labs. We have also been used at the bottom of the ocean. We've been used in the International Space Station. We've been used in remote parts of Africa. We really pride ourselves on being very dynamic technology. We really want to be able to serve all types of people as best as we can. So, about 20 years ago was the first whole human genome. We had about 15 years of Sanger sequencing, 15 years of sequencing by synthesis, sort of the shorter read sequencing. But there was a study in about 2022 that said that about 20% that has been done, 8% of the remaining is completely unknown. And of that 8%, there's a lot that Nanopore can resolve. And we have a belief now that what you're missing really matters. So what is Nanopore Technologies? So this right here is a nanopore. What works is we have a synthetic membrane that has a whole bunch of these blue pores in it. We have a motor protein that will attach to the spore and run native, so not modified, PCR-free, if you'd like, DNA or RNA through this pore. When it gets run through the pore, you'll have an electrical gradient that comes out, and this gets directly translated into not just your base pairs, but also the modification, so all of your methylation. I was asked this a few times today, like, can we get methylation with your product? How do you do it? For us, it is the exact same sample preps, exact same library prep. You don't do anything additional. The only thing that you change is on the computer when running your software, you click one button and it will come alongside everything else. So very simple and very comprehensive. So direct sequencing DNA and RNA. This is really important. When you PCR, which is necessary for some samples, you are taking that DNA or RNA and you're ironing that out. You're taking away all of the modifications. And we now know how important those modifications are. Epigenetics, structural variations, all of that's very important. We are amplification bias free, so you're not getting artificial amplification of certain regions that make it look like you have a higher copy number variation. We are read length agnostic. This is a big one. So traditionally we're seen as a long read technology. We are capable of long reads, we are great at long reads, but we're also short read capable too and everything in between. The shortest is 20 base pairs now, which is liquid biopsy level. The longest 4 megabases. So we can cover whole genomes or we can cover up to very short bits depending on what you're looking at. And then real-time analysis. For people who work in labs, this is incredibly important. I'm going to add batch free here as well after this, but the real-time nature means that when something's running through that port, that blue line that comes through, you see that as that happens, which means that if something goes wrong, you're able to stop your run and reload and rerun that and fix that straight away without losing consumables, without wasting money. But also, if you reach the depth that you want, you have the result that you need, you are able to stop that run, take out, wash that cell, and load a new sample onto that, which is really important for being economical, saving money, getting the maximum out. So the way we work is we have flow cells, which get inserted into our devices. They have a certain amount of data you can get out of them. And you can sequence and wash until you use all of that data. So what can we be used for? SNVs, structural variation, epigenetics, phasing, gene expression and single cell, fusion transcripts and special genomics, splice variation. We're really used across the board. And what we say is everything that runs through the pore we can sequence. So we are, as with single cell, as with spatial genomics, we are able to see quite a lot down to the very small minutia level. So, sample prep, another thing I've been asked about a lot today. When you're comparing us to other platforms out there, what is one of the main differences? Things about sample prep, instead of taking about 3 days where you're seeing that average, our longest whole clinical human genome is about 4 hours, and our shortest rapid is 10 minutes. That means the human time that's necessary and the training is much, much less, which means that your lab techs, the people that are working on that, have more time to do other things. It makes it a lot more functional. So scalability. Our smallest device, our MinION, which is at the booth right now, is about this big. So think a large USB. Our largest device is a large printer. So we're not talking about the 1980s computers that are filling half of a room. These are portable, these are dynamic. Our MinION, this tiny one, is point of care applicable. It's used again in remote parts of Africa. It's used in the jungles. It's used at bedsides where it's necessary. For places that are doing larger sequencing, we have our GridION, which is 5 MinIONs. And anytime you're doing whole human genome or whole genome at all, we tend to move towards our Promethean line, which the smallest is a P2. The number is a big number of flow cells, so the number of things we can run. And I was going to touch on this anyways. It's debatched, which means that every single one of our flow cells, every single one of the ports runs fully independently. So if you're running, say, a clinical lab, and you have a sample that comes in now and one that comes in 5 hours and one the next day, you don't have to wait 3 or 4 weeks to get enough samples to make it economical for your run. You run them without wasting consumables just for what you need to do, and you can have run different protocols. Again, saving money, saving time, faster turnaround times, and particularly when you're dealing with patients, it's really important. And the last part here, we are able to be no CapEx. So talking about price, talking about things like that, I'm not going to say too much on this, but you have the ability to buy an instrument. And when we talk about the price per genome, we don't often account for that in the run. So instead of being, say, $500,000, $1 million upfront, we have the ability to have our starter packs, which means that we're loaning our instrument out to you and you're just using it on a consumable basis. This means that you can start sequencing, start researching without maybe that huge upfront grant. That's really important. It's been important across the board. And that's available not just on our huge devices, but even our small ones. So we really have tried to make this as accessible to as many people as possible. So back to the genome. Mayo Clinic said about 8% of the genome is missed by sequencing by synthesis or short-read sequencing technologies. 20% almost of the human genome is inaccessible to short-read sequencing. Decode found that 40% of structural variation tends to be missed. 3% of CPG sites by Exeter are completely not able to be found. 34% of disease-causing variation from University of Washington were unable to be found by single base pair substitutions. And there is a huge amount, about 50% by Seattle's children, that said we're not able to diagnose. When we're looking at negative exomes even, we're finding a lot of people are not being properly served. There's a lot of long patient odysseys, and we're trying to shorten that. We're trying to change that. We have a lot of cases where we've been able to take negative exomes, people who've been on long patient odysseys, and actually resolve those, this year included. So we're trying to change the whole system as it goes. And what you're missing really matters. All of this missed information goes towards what you treat the patient with, what you can identify them with, what disease they have, or even when you're going down to the microbe level, what kind of resistances they have. It's all really important, and we have more and more examples every day, every year about this. So here, this is just to show that nanopore is really good at identifying complex variation and complex regions. So here's an MHC, where you can see a comparative. And then over here, SNV identification. Nanopore is very good at figuring these out. These are important for the range of items that we go through. MHC is one of them, but this is also things like HLA, and then up here is things like thalassemia. So here's CPG sites. We see about 3% of CPG in, say, an EPIC array. Nanopore can get 93% of this. This is really important to a lot of diseases, including things like hereditary cancers, but also things like brain cancers, breast cancer, and the rest of the oncology scene alongside other parts of clinical diagnostics. So here we have an example from Julian Maslaya from Marie Curie Institute. This is the multimodality of nanopore, showing that they believe, particularly with things like adaptive sampling, which is one of our cool techs I'll go over right after the slide, methylation, structural variation, gene fusion, single gene mutations, and phasing are all super important. And nanopore is very good at picking up all of these levels, and is thus going to be introduced to clinical routine practice in Europe at Marie Curie Institute. So what is adaptive sampling? This is one of the coolest bits of our technology that has come out in the last year or so. So what happens here is with just a BAM file, you're able to identify regions you want to artificially or bioinformatically enrich or deplete. This means that you do not have to do that ironing or modifications that you might otherwise have to have. And you can have as many target areas as you'd like, which is also very important. So what happens is it will read, our pore will read the first, say, 100 or so base pairs and go, is this the target sequence we want? If it is, you get the bottom, runs through, it gets sequenced. If it's not, this gets ejected out, and that's part of the sequence is not read. What that looks like in practicality on this hereditary cancer panel, here every bar is a specific gene. The Y is a graph of depth, and it's over time, so this is growing over time. So you can see an equal coverage over all of these genes, which is very important first off. And the 40 or 50x clinical coverage that we're often looking for is actually surpassed. We're averaging over 60x depth. The other two important things about this is looking at, again, hereditary cancer, BRCA1, BRCA2. You can see shoulder regions and main target region that are amplified, where you have, I'm gonna walk over to the point, sort of this area and the other one, where you can see the enrichment of what you want to see, that depth that you want to see, and everywhere that you don't want to see in the middle is really depleted. What you get with this depletion is still a low pass full genome, which can be very, very useful if you want to look at a different area, but a very helpful artificial enrichment. Okay, comprehensive results. So this is more of the stuff that we've done with adaptive sampling. We've been able to resolve inaccessible regions. This is stuff for hemoglobinopathies, which I've been asked about today, things like thalassemia, really important to get those intractable regions and intractable diseases. The all-in-one assays, everything can be done on our devices, and you can choose different applications, direct RNA, direct DNA, methylation, all of the sites. It's you're able to see, again, phasing in haplotypes without having to run parent samples. Again, if you're trying to save money, that's very important. And then it's a very fast, rapid turnaround time. Earlier on, I talked about that 8% of the genome that's missed. This has been termed the so-called dark region or dark genome by Dr. Ebert. Of that 8% that is missed by most technologies, we can get already, and this number is changing, already 90.4% of, again, very important clinical regions. This is where things like SMN1 and SMN2 and CR1 are really important for rare disease that are very prevalent in the region. For STRs, so this is a repeat expansion disorder. Because we aren't chopping up so much as many other technologies might do, we're able to see things like repeat expansions to much greater clarity. So here we were able to look at 50 diseases with one test with unusually long repetitive DNA sequences and STR expansions. Again, things that are usually very hard to sequence and bioinformatically programmed. One example of where this was really useful, there was a patient that had a 10-year diagnostic odyssey. They didn't know what the diagnosis was for 10 years. And they were able to resolve this case with Stevanovsky et al. In 2022, so last year, ending again a 10-year patient odyssey with a patient with Canvas. So this has real-world, real-time applications. CNV analysis for neurodevelopment disorders. Adaptive sampling has been used to sequence over 30, and this is just showing that we had 100% accuracy with the relevant CNVs, or copy number variants, and the variation. So also here, what's noted in the graph to the right for you guys is haplotype 1 and 2. You can see the haplotypes, and up top is the methylation. All really important information for when you're trying to treat patients. So actual and accurate information in real time. So here, in under 8 hours, they were able to derive candidate variants. In under 2 hours, they were able to do a whole nanopore genome. We actually have the world record, Guinness Book of World Records, for the fastest clinical genome where it's necessary. And while that might not be necessary for all clinical settings, it does show you how advanced and how quick we can do things. And where it is required, we are able to speed up and match capacity. So this was an example with a 3-month-old with epileptic seizures. So you have clinical presentation, and they were able to identify the pathogenic variant within 8 hours. And it was a Poirier-Benvenu neurodevelopmental syndrome, or POVINES. They were able to stop, change, and completely alter the treatment for this patient. Again, this is sometimes a life-changing difference. And the standard of care test that they did at the same time, what was currently used, came back 2 weeks later. Again, that can be quite a lot of time for when you have sometimes very serious disease. This is another example where we had a 3-hour genome sequence of variant. So there was a newborn where the older sibling had presented with a clinical presentation shown in the blue arrow over there. And within 8 hours of life, they were able to not only figure out that the newborn was not a carrier, but also able to identify that he was not doing any of the disease or presenting any of the disease and would not. So for parents, for patients, for all this sort of stuff, the turnaround time really matters, the accuracy really matters, and this is, again, life-changing differences. So one last thing for me to touch on, because I know I've been mainly doing clinical presentations, just quickly on some of our metagenomics and some of our respiratory clinical metagenomics. We are doing an early access program, including here in Pakistan, but around the world for TB, which we know is a worldwide problem, a worldwide issue. So for our drug resistance testing, some big numbers to point this out, from sample to report in 6 hours or less. So very quick. The sequencing is only 2 hours. 16 anti-TB drugs screened, 1 integrated solution. So all of this can be done on our GridION and is what we call our Q line. Q line is something that will not be changed. Our technology evolves very quickly. We understand we used to have an accuracy. Our accuracy is now 99.9 or 99.997, depending on where you're at. It used to be much less a few years ago. We evolved very quickly, but we know for some clinical settings, that doesn't always suit. So we developed our Q line so that things will stay stable for over a year, so that when you're developing a test or using a test, that can be stable. And this is one of the things developed on that Q line product. How this works. One major thing, this is just decontaminated sputum. So it's fairly direct from sample. Decontaminated sputum and cell lysis, extraction, multiplex PCR, rapid barcoding, sequencing, analysis, and report. Again, 6 hours. This is, we found to be a game-changing result. We are hoping to do early access in Pakistan, among other places like across Europe, in Saudi Arabia, in the UAE. One thing about pricing, I know, again, this comes up, how do we reduce price? Multiplexing is one of the things that we do in barcoding. So for samples like this, we are able to barcode, depending on the type of sample, 12, 24, 48, or even 96 samples per an individual flow cell, which means that if you have a lot of cases coming through and you're trying to reduce costs, that's one way to really do that. And we found that to be really important. Again, we want to make sure that we can fit and suit the markets, the people. We want to help as many people as we can. And so we're trying to find solutions to do that. And TV is one of the great examples. That early access is now, and then in a few months after that, hopefully more generalized access. So nanopore, just to summarize, comprehensive insights, any fragment length, ease of use, real-time and rapid, scalable, economical. And honestly, I'd like to say things like approachable. It's easy to learn, it's easy to teach, it's easy to expand out on. And we're using this to try to help as many people as possible. And I will note, this is the human aspect of mainly talking about a little bit of metagenomics and respiratory metagenomics. We do work, obviously, in agriculture. We do work in conservation biology. We're in a lot of disease surveillance. There's a lot of other areas. So this is just touching on part of our more clinical aspects. But I appreciate you guys all listening to me today. And thank you guys for all your time.
Thank you very much, Casey. This was a really nice talk. Now I would like to invite Dr. Faisal Khan.
Just one, we are running late, so we'll allow one question. Please,
Could you introduce yourself? That would be great.
Yeah, this is Dr. Osman, the head of Basic Sciences Research of Khan Cancer Hospital. So, Catherine, very interesting talk. So, I have a basic question about the identification of large deletion or duplications. So, how large deletion, up to 5 or 25 kb, could be identified through your technology? Could you please comment on this? Thank you.
So I'll respond more. Sorry, I can't really see you directly, but we've had researchers around the world who've done incredibly large deletions. I can send you also direct papers about this. We have been able to resolve sort of complex deletions to very large fragment lengths. I do not remember the exact length, but it was more like 4 times. So if you don't have that, or add me on LinkedIn. I would love to answer your questions. I'm really nearby on flights. If you'd like to come say hi, or I'll be back maybe sometime in Pakistan as well. And I have my colleague Osman as well, who's here to answer questions. But there's a lot we can do. We hope there's a lot we can do to help and support Pakistan as well. So
thank you very much.
Can we identify both methylated and non-methylated nucleotides by this technique at the same time?
Yes, yes you can.
Okay,
thank you.
Thank you to the moderators.
So,
thank you everyone.
I would like to invite Dr. Faisal Khan. His talk is on precision medicine in Pakistan. Dr. Khan is a principal investigator at the National Center Big Data and Cloud Computing Precision Medicine Lab in Oman Medical Institute, Peshawar. So, please welcome Dr. Faisal Khan. Dr. Khan has his PhD from the University of
Thank you. It's an absolute pleasure to be here. I'm really grateful to the chairs and the organizing committee, especially Dr. Natasha, for the invite. I think we're short of time, but my job here is to give you a glimpse of what we're up to in Peshawar at the Precision Medicine Lab. I'll be sharing some vignettes from the lab, and I hope it sparks a discussion over the course of today and tomorrow. So these are some disclosures. So I'm a biologist. We are a biology lab, a very proud interdisciplinary and translational biology lab. But I'll only be covering the medical side of stuff that we're doing. But as you can see, it's very broad, and we touch all sorts of different sectors using all sorts of different tools. So quick history, the Precision Medicine Lab is jointly run by CCOS University and Rahman Medical in Peshawar. It's a node from the National Center for Big Data and Cloud Computing. This was a ₹1 billion project back in 2018 by the Planning Commission, and we're leading the medical side of it. We sit at this really dense cluster of healthcare, a 1 square kilometer with a huge sort of density relative to the rest of the country. And that's where we benefit from, both in terms of patient samples as well as talent. And again, so we're very interdisciplinary. The team on any given day is around 20 to 30 people coming from all sorts of background, sitting together, cracking at the same problems, using their different languages and different tools. We have a strong bias towards prototypes, so we're really keen on solving problems that we're surrounded by in different sectors. On the medical front, the mission is the following: to glean every single data point available to optimize cancer treatment and minimize ineffective treatment to help every cancer patient and their household. And we're constantly learning, and we've been learning over the past 5 years, and I'll be touching on some of those. So we built a very central lab at the pathology department in Rahman Medical from scratch. This was commissioned in 2018. And these are some vignettes from what we're up to. So these are some areas that we're touching on. Different groups have been working on different layers of the problems, and I'll explain how this comes together in our oral cancer work in the next few slides. So I don't think I have enough time for each one of these, but just to give you a taste, we've been looking at miRNA markers within head and neck squamous cell carcinomas, and Laiba and the team has been able to narrow down on 5 different candidates that we're now looking deeper into it from a local population perspective. We also do a lot of network biology. So in oropharyngeal cancer, we know HPV is a main culprit. So we do virus host networks, protein interaction networks. Arsalan has been doing it, and this work was presented at the American Association of Cancer Research meeting last year. So basically, we identified several interesting clusters that were common between OSCC and OBSCC, oropharyngeal and oral squamous cell carcinomas. And that's what we're trying to validate back in the lab now. On the microbiome front, we're also sampling our main cohort of patients for their salivas, which means we'll be looking at the oral microbiome. But Abdul Salam in the lab went out and did a meta-analysis of what's out there. So if you can see the red bar, we think Fusobacterium has a role to play, but we're very keen on what comes out of our own microbiome data in the next quarter. We also have a digital pathology team, which is developing a low-cost solution for local hospitals. So we don't want a gigabyte of one image posing lots of storage problems in the hospital. We don't have a lot of compute at every hospital. So we said, why not repurpose existing microscopes and develop algorithms which could help us do some predictive work. So we're currently able to stage esophageal cancer. But we recently won a grant from the HEC to expand this and benchmark it against whole slide images in 4 other cancers as well. So this is getting very exciting. Again, Professor Iqbal, head of histopathology, has been instrumental, and his team, of course, because labeling is a key issue, is a time-consuming part of all AI experiments. And this work was presented in these 2 conferences as well. We did a very exciting wearables problem experiment within the team, not going for the famous Apple Watch, but we went for Mi Bands. So these are 6,000 rupees bands that are very sort of common in the local market. and we thought why not use this to track some candidates. We layered with other self-reported data and some vitals, and we were able to predict different parameters of the body. And this was an award-winning poster at the ISMB. Now what we're trying to do with this, just to give you an idea of how we scale, so we were speaking to gynae people now to scale this up in first-time mothers, tracking them for like a 10-month period to see what we could predict in terms of hypertension, and also, for example, a big problem is stillbirths. So all those labels combined with the data coming out from E-bands. Again, and layering it with other data sets as well as their ultrasound scans. So we don't let go with the data that's based at the hospital. So this was a very exciting hypothesis coming out of COVID conversations in the lab. We were desperate, we wanted to do something. So again, Saddam came up with the idea of a molecule. We'll call it a molecule X here. And what we did was, the hypothesis was it might be having some predictive power, and it should come out in the urine. So we went back, we just emailed our IT department, and we said, can we have urine results of all COVID positive patients from the past 6 months? And we saw some difference, and that's what we're trying to hash out now. So again, some of your hypotheses could very quickly be validated if your hospital's keeping medical records electronically. And then one of my favorite ones as a biologist, of course, is the wet lab, the cell culture lab. So all our patients, we sampled them for the tumor DNA, for the tumor aliquots, for blood and saliva. And of course, a very detailed patient history. That's where all the labels come from. But we also take some tumor aliquots into the lab within a 30-minute window, and we expand them into cell lines. So these are primary patient-derived cell lines, which are very useful for taking your predictive results back to the lab to be able to validate them. So we have a growing repository of primary cell lines from oral squamous cell carcinoma patients. So what's the oral cancer genomics pilot about, and how it's evolved from a simple genomic project to a more multi-omic one, as I'll explain. So quickly, I'll just have to breeze through this. Again, oral cancer is very huge. You see all cancers, mortality versus incidence, 2018, both sexes, all ages. But if you narrow down lip and oral cavity, Pakistan goes at the top. So you see the purple dot there. So the subcontinent is pretty much the capital of oral cancer in the world. And that's why we chose this for the federal project. Again, no time to go into the molecular biology of it, but it's very heterogeneous. It's very poorly studied, although it's very important from a local perspective. And it has its own biases. So I'll quickly come to this. If you see most samples are HPV negative, which presents a bias there and doesn't let you explore all the HPV positive patients. And also the data is predominantly white. So, and we see these undercurrents of disparities when it comes to representing different populations. And that's what we're trying to beat with our project, is to decompose some of the heterogeneity and understand it. Hopefully be able to detect HPV. There's a paper on EBV being a major cause, potential trigger for cancer initiation in Asian populations. So we'll be screening for both. And of course, we have a predominantly Pakistani population. So we'll see what the signatures are like when the results come out next month. Just to give you an overview, it started off as an exome project. We quickly were able to integrate all the other layers of data sets. And from an AI, from an engineer perspective, becomes a very exciting playground. But then we were like, why not sample preemptively for all the other layers? And we were lucky to have been able to crack some of them now. And we're looking for partners who can help us unlock the ones which are not there yet. And as I said, it has a whole biorepository at the back, including their primary cell lines. Again, from an AI perspective, if you go forward, we've been trying to be very comprehensive with the patient history forms that we have, and not just looking at stages or risk factors. We're also going deeper than that. There's a food paper there from Iran which says achar could be a risk factor for oral squamous cell carcinoma. So we've even included that in our form to see how it goes in our population. So I'll close with this. We're keen on clinical services. Remember, we're tools people, we're biologists. We're not in love with any specific disease yet. So we want to solve as many as we can. So just to give you what's to come, we're working very closely with Rahman Medical, and we've just taken over the Center for Genomic Sciences, where we're working on oncology, genomic testing in oncology. Neonates are second. That's one area we're expanding pretty soon. And another one's very fascinating, which I'll explain in the next slide. But we talk about genomics, but it's been a year now in this journey, but we quickly realized there's a huge awareness problem. And there's a huge reporting problem. And not every clinician or clinic is ready for this. So it kind of did slow us down. We quickly went out, did different surveys, which will soon be published, to understand how much our clinicians are able to decipher the reports, what do they understand and what not, and also what's the general genomic literacy rate out there in consultants and trainees at the same time, hoping that this will make a case for better training and newer programs that could help us create a critical mass of people. From the wet lab, we have a cancer cell culture as a service proposition here. Since we can draw cell lines from the patient's tumor, we're able to do drug response assays within a 2 weeks time period. And these are some of our preliminary results which we shared at a MDT at Harvard teaching. So as you can see, the tumor cells from the patient tumor are pretty much resistant to some of those chemotherapies out there in practice. And again, we're exploring this closely with breast cancer surgeons as well. So again, being able to do drug assays and share those results within a 3-week window, hoping to improve treatment outcomes. I just want to wrap up with this slide. We need each other. We need large-scale data sets in order to leverage all the technologies that we heard about. Great stuff coming out on that, and how do we deploy them in the Pakistani context? Again, 4th or 5th largest country in the world. Not many people are aware of this. A huge opportunity out there, but how do we create these large-scale data sets together, representing the population that could help us, say, develop panels and bring the cost down for the patient? We have to be aware of patient realities out there. Plus, there's a huge awareness piece that needs to be resolved, and we can only do that together. I'm really excited to hear about the molecular society out there coming up. I don't know how we can work together, but we should. There's a capacity piece too. For example, the province of KP has 5,000 biotechnology graduates, according to one study we commissioned. How many are genomics ready is the question. And then how many TMOs around you are genomics ready? The consultants of the future, right? So again, all these companies, societies, doctors, biologists, bioinformaticians, we need to come together and crack at this sooner than later. This is not going to happen organically or by default. And we don't have time to wait for this. So I really hope we can catalyze this somehow. And I'm just left with thanking all the clinical teams out there, without whom this wouldn't have been possible. And of course, the team in the lab. Some of these are elective students, MBBS and BDS elective students. Really want to point that out. Took us very long to offer an elective in precision medicine to these students. And many of these are authors and presenters at the recent Shaukat Khanum Cancer Symposium. So it's a fabulous accomplishment at their age, but just to kind of demonstrate the potential out there in our youth that really needs to be harnessed. So yeah, please like us, follow us on socials, visit us, and join us in this movement. Thank you.
Thank you very much, sir. This was a really wonderful talk. Our next speakers will present their talk in 5 minutes.
Will we take questions before the pre-papers? Dr. Sam, if we do this? Because we're like about 25 minutes behind schedule right now. So we can talk. Yeah.
If our next speakers can manage the time, we can take questions.
Speakers से request करेंगे. Dr. Faisal, I hope you will stay till the end. So because we would like to have a question answer session. Sure. And other speakers also.
Yes. And we can discuss on the tea as well.
Okay, so we've had a really great inauguration of session 1. We've learned about a lot of the technology that is out there, some of the fantastic work that's going on in precision medicine. And now we're going to hear about some of the other institutes that are also working with next generation sequencing. So the pre-paper session is going to start now, and we're going to ask the presenters, please, you have 10 minutes. That 10 minutes includes 2 to 3 minutes for question answers. Thank you.
I think we have 5 minutes. For each speaker now, we can manage. Well, it will go to 40. Yes. First of all, I would like to invite Mr. Ali Islam, who is the lab supervisor. Senior medical technologist, molecular pathology, Department of Pathology.
Thank you, Dr. Salah. Assalamu alaikum and good afternoon, ladies and gentlemen. I'm Ali Islam from Molecular Pathology of Shaukat Khan Memorial Cancer Hospital and Research Center, and I have been privileged to present the first ever oncology NGS data from the Pakistan, and the fact being that all of the genomic studies and the precision medicine data that has been accumulated across the globe is mostly based upon the Caucasian and the European population. So it becomes the key, it's become mandatory to analyze the genomic profiles of Pakistani individuals to screen out the marker of sensitivity and the marker of resistance, specifically in the cancer patients. So in context to my presentation, so let me take you through the NGS that basically allows you to permit the sequence many genes simultaneously, that allows the identification of many disease-causing mutations that may be CNVs, SNVs, insertion, deletions, and etc., thus giving you a genome-specific profile to target all those clinically actionable variants. So let me take you through the World Economic Forum that is completely an autonomous but non-clinical quantity that has realized the importance and potential of NGS in precision medicine, that it has declared and aspired the precision oncology as the best new weapon to defeat the cancer. So based upon the NGS data, the FDA has given the accelerated approval for NGS-based assays to the actionable cancer genomic aberrations. And followed by that, the applications of NGS are not only in the clinical oncology, but that's also in clinical genomics, high-resolution transplantation typing, rare genetic disorders, precision medicine, and so on. So related to that, let me take you through the very complex workflow of the next generation sequencing, in which an FFPE or the blood specimen is histopathologically reviewed to mark the tumor content. That is followed by the nucleic acid extraction, and ultimately the library preparation, in which each DNA amplicon is given a specific index, and that's an EGFR to be negative. And finally, when the NGS performed on the same sample, it has shown the very surprising results that was mapped for pathogenic variants. two frame shift deletions at the same time, that was unable and beyond the resolution power of conventional assays that include RT-PCR and PTEN mutations that were likely pathogenic. So as a clinical consequence, the SMAD4 deletions are a poor prognosis as they promote independent tumor formation and accelerates metastasis. So here we have the other case where a 25 years male was presented with a brain tumor. So molecular testing has shown it to be the 1PQ19 positive for FISH and IDH1 mutant glioma. So when the NGS was performed on the same specimen, we have found the very astonishing results that has shown that the singleplex assays are not much enough to completely and comprehensively screen the genomic aberrations that are present in that tumor. So in addition to the IDH1 pathogenic variants, we found KRAS variants that are very classical G12A mutations were pathogenic, and TP53 pathogenic variants were also identified. So meaning thereby, so singleplex and conventional assays are not that much enough to completely resolve the genomic signatures of the tumor. So here are some other cases, and finally this is the report that how do we report the NGS data, and finally the variant interpretation as per ACMG guidelines, and this is the percentage of the tumor that we receive in our lab, that is the breast and ovarian highest, lung cancer by 26%, and other and GIT cancer. And this is the relative distribution of the variants that we found, the KRAS and TP53 variants at their peak. And this is the 50 gene panel we are performing in our lab. And now we're looking forward to expand further, where we can have a 70 gene panel where the genes in the orange circle that have been additionally added to this existing panel. And you can also have the RNA fusion partners and CNVs with the same panel, and you don't need to perform the separate assays on FISH and RT-PCR that include ALK, fusion, ROS, NTRK, MET14 exon 19 skipping, and finally the landscape of molecular pathology, where we have start performing singleplex assays in 2013-14, followed by KRAS and IDH mutation typing, and finally BRCA2 and pre-transplantation that were all singleplex assays. And now, 2023 onward, we have started performing solid tumor panel sequencing, BRCA1-2 genetic testing, and finally the myeloid panel. So there is a paradigm shift from single gene testing to the multi-gene testing because the singleplex assays that are not enough to completely resolve the tumor-specific signatures. And so, to take toward the complementary and other kind of medicine. So in the summary, so NGS is a robust and powerful technique for interrogation to give the clinically and drug response association. And there's a paradigm shift from single gene to multi-gene testing, and it's undoubtedly saved the precious sample. And at the same time, it leads to the precision and personalized medicine, so that relies upon the right drug at the right dose to the right patient at the right time. And I'm greatly thankful to Dr. Ramina Ghazi, my section head, for her tireless effort, and to the senior management of my hospital, especially Dr. Faisal Sultan, Asif Loya, and Dr. Asim, for their tireless efforts. And I'm grateful to you all. Thank you very much.
Please, one question can be asked.
So that was really wonderful work you have presented. Just one short question: that how you narrow down these genes? You did whole exome or some kind of
Thank you, Dr. Israr. So these panels have already been validated by doing 100,000 of genomes across the globe, and this panel is Illumina validated. So all of the genes that have been included in this pan cancer panel that have a close association to the cancer. However, we have only the hotspot regions and the hotspot genes. And now as the data is exploding, now we came to know that only a 50 gene panel is not enough for the comprehensive genomic screening. Now we are shifting to 70 and 100 so that we can have a more comprehensive profile. So this is the very basic start, and this is the landmark we're going to establish more in our lab, and this is the very beginning. So most of them are relevant to the hotspot regions in the cancer.
Now I would like to invite our next speaker. The topic of the speaker is uncovering genetic variants linked to non-alcoholic fatty liver disease. Dr. Asma Shabir will present from Pathology University. Will present her research.
5 minutes? Shall I try?
5-6 minutes, then we can have one question.
Assalamu alaikum everyone and a very good afternoon. I'm Dr. Asma Shabir, PhD scholar from Ziauddin University and Assistant Professor at SMC Jinnah and Medical University, Karachi. The research topic is uncovering genetic variants linked to NAFLD in Pakistani population, insights from a whole exome sequencing investigation. This study is a part of my PhD project. Well, as we all know, non-alcoholic fatty liver disease is excess accumulation of fat more than 5% in the liver. It has a spectrum with steatosis, it added with inflammation as NASH, then cirrhosis. NFL is the most common cause of chronic liver disease and has become with a high related morbidity and mortality. It is alarming to know that there is 50.4% increase in prevalence of NAFLD over about 3 decades. So this is a matter of concern where NAFLD can lead to serious complications like cirrhosis and in fact hepatocellular carcinomas. It is a multifactorial disease and genetic elements are widely studied throughout the world. But there are negligible studies from Pakistan. Hence, we aim to investigate genetic variants associated with NAFLD in Pakistani population using cutting-edge whole exome sequencing combined with bioinformatic analysis. For this, the methodology. This cross-sectional prospective study was conducted at Ziauddin University. All the patients were recruited at GIT clinic. After taking informed consent, detailed history was taken and patients were physically examined, followed by primary screening by ultrasound. Patients with hepatic steatosis were included and subjected for various biochemical tests, followed by transient elastography, which identified the grade of the steatosis and stage of the fibrosis. Considering all inclusion and exclusion criteria, we determined a cohort of 200 cases and then was followed by a case control study. 6 cases were selected from this 200 cohort and 6 controls were identified from healthy population. Total 12 participants, blood was drawn from them and then subjected for DNA extraction by using QIAGEN kit. Then whole exome sequencing was performed using Illumina platform. Bioinformatic analysis was done and various tools were used, namely like Burrow wheel aligner, Picard tools, STIPF and GATK tools. Then, according to the ClinVar database and following the ACMG criteria, American College of Medical Genomics, we identified the variants which were benign, pathogenic, variants of uncertain significance, lightly benign and lightly pathogenic. And SPSS was used for calculation using SPSS version 26. Then we came out with the results with the baseline characteristics of these 200 cohort where male were predominant with high BMI and most of the patients were hypertensive, diabetic and had dyslipidemia with elevated ALT and AST enzymes. The whole exome sequencing data of these 12 participants were jotted down for their mutations and frameshifts and indels. The most interesting part, by the ClinVar analysis, we identified the 3 pathogenic genes in our population associated with NAFLD, namely SUMO4, PRKCQ, and MBL2. We noted SUMO4 was found in 83.3% of cases and 50% of controls, whereas the other 2 genes were equally seen in both cases and controls. So a very interesting finding was noted further, where we found a significant association of SUMO4 polymorphism with diabetes with p-value less than 0.05. So basically, SUMO4 is a cytoplasmic protein known to be involved in post-translational modification. And it is also a regulator of nuclear factor kappa B. And literature shows that SUMO4 polymorphism is associated with the susceptibility of diabetes. And similarly, in our study, we found that 5 out of 6 NFLD cases were diabetic with SOMO4 polymorphism. And NFLD and diabetes are closely linked to each other, hence suggesting that SOMO4 variants may contribute to increase the risk of NAFLD in our population. So we conclude that the strong association between SOMO4 and diabetes contribute to the development of NAFLD in a subset of population, and our study provides valuable insights into the genetic underpinnings of NAFLD in the Pakistani population. So here comes the challenge to the physician, where the diagnosis of NAFLD is very challenging. So getting the information of this genetic piece, which is an important piece of the puzzle, understanding this genetic underpinnings of NAFLD is very important, and a thought must be given for this in order to uncover the precision medicine in future. And these are my supervisors. I am grateful to them. Thank you so much.
Thank you very much. I would just like to make a comment that I think for determining an association, the sample size should be much larger. So
yes.
You can suggest, but you cannot conclude it.
Yes, I definitely agree with you, sir. As I told you, this is just a part of my PhD project, and that's why we collected the 200 cohort samples. And this is just a small part showing from the ClinVar database. We are further analyzing using different prediction tools, and then further we will validate our study among these 200 cases as well using Sanger sequencing, and much to go ahead. This is just a piece of that study.
Thank you.
When I have one question that you mentioned that 83% cases are positive for SMO variants. Thank you very much for a wonderful talk. I just want to mention that SMO4 data you mentioned that 83% cases were positive for that variant and 50% control. Don't you feel it is quite non-specific if you want to relate this variant with the disease?
Well, when we compare with them, like when you compare the cases and controls, we have 83% like higher rates in cases as comparative to control, right? And this SOMO4 was predominantly in diabetics as well. Further, like controls we do have, but what we thought that they are having this mutation 50%, but we are thinking to follow up them for the further 1 year and see about their clinical conditions. At present, they are non-diabetic. Controls are all non-diabetics, right?
Because the number of samples is too low, because it means 50% means 3 out of 6 were positive for that variant in case of control.
Yes.
And 4 out of 6 or 5 out of 6 were positive for that. So this is very minor difference, and I think it should not be related with the disease.
Yes, this is not.
I think we need to stop at this point. You can have a discussion later on,
sure,
because we really need to move to the next speaker. Thank you very much. Thank you.
Thank you very much. Our next speaker is Dr. Fazala Zafar from Aga Khan University. She will present the research project.
So, Dr. Ghazala, you have like 6 minutes. Okay. Please, I know it's unfair to tell people at this time, but we're just running short.
It is better to discuss results.
Assalamu alaikum everyone, this is Lalana Zafar from Aga Khan University. I'm working as a senior research assistant in Aga Khan University. Grappling with the challenges of rare genetic variants, today I'm going to present our study on a novel homozygous frameshift variant in SPDBN4 gene causes axonal neuropathy with intellectual disability in a continuous Pakistani family. So, our study started when a couple are walking to Aga Khan University facing the challenges of neuropathy, deafness, and ataxia. So, this is the pedigree drawn by us of the family. As you can see, here both of the parents are first cousins, and 3 of their children are affected. So this is the clinical. These are the clinical symptoms of our patient. They are having a developmental delay, congenital hypotonia, neuromuscular weakness, loss of deep tendon reflexes, epilepsy, cortical visual impairment, respiratory difficulties, feeding difficulties, and neuromuscular weaknesses. So this is the pictorial representation of the patients. So our objective is to identify genetic causes that leads to rare disorders in coterminous Pakistani families. As all of you know that the rate of coterminous family is coterminous marriages is very high in our population. So which leads to these disorders in our population. So I'll go through with the methodology fastly. So first we recruited family recruited, and then consent form were taken from this patient, from the family, and then blood collection, DNA extraction, whole exome sequencing were performed, then data analysis by different in silico tools, and then candidate variant were identified. Then for further confirmation of our variants, we did Sanger sequencing analysis. So I'll go to the results. After getting data from whole exome sequencing, we use different in silico prediction tools that either our finding is pathogenic or not. So we use SMG criteria. So according to SMG criteria, the variant we got is pathogenic. So these are the findings of classification of our results. So Sanger analysis. As you can see here, the parents are heterozygous mutant, while the parents are heterozygous mutant, while the affected individual are having the homozygous mutation in this region. If you can see here in the homozygous mutant, there is a deletion of G and C in both of the alleles, while the parents having heterozygosity, having the mutation in only one allele. In this portion, you can see here there is a deletion of G and C base pair. While this is a wild type chromatogram showing there is no deletion of G and C. So I will go quickly with the SPTBN gene. Basically, this gene is a member of family of beta spectrins. SPTBN gene encodes the beta 4 spectrin proteins. They plays a vital role in stabilization and organization of neuron, which is predominantly expressed in central nervous system. So this gene is 2,556 containing amino acid, and it's a very large gene. Due to mutation in the position base pair number 1799 position, there is a deletion of G and C. Due to that, this is the wild type protein structure, while the mutant protein showing the truncation of the protein and due to all this of codon. So, as the prevalence of this disease is still unknown, but there are several variants reported up to date. Up to date, there are only 20 variants were reported in this gene. So this is the summary of all the variants. Including our variant, there are only 20 variants. 13 of them are truncated mutation, 3 splice-site mutation, and 5 missense mutation. And this is our study. So this is the final conclusion that the novel variant in SPTBN gene having deletion in 1799 position is causing this disease. So our current study expanded the mutation spectrum of SPTBN gene in association with neurodevelopmental disorder. So identification of pathogenic variant will standardize disease prevention by genetic counseling to the family. These are some references. Thank you so much.
Only one question may be asked, please.
So this was already reported.
I'm sorry, that lady raised her hand before. Go ahead.
Was as you did the exome sequencing, so was it the only mutation that you find and suggested it as a causal reason for the neurodevelopmental disorder, as there are already many mutations which are considered as the causal and also they run in families as well. So how did you conclude it from the whole exome sequencing that this is the one?
When you read the whole exome sequencing, you get so many variants in different genes. So for that, you have to filter out your data with the phenotypes you get. So we filter out those data, and then, as I told you, according to SMG criteria, either our variant is causing the disease or not, then we will go for the SMG criteria, and this was the novel mutation we got, and we reported this.
So this can be one of them.
Yeah,
besides the other one. So this is not the only.
This is not the only one. This is from the only novel mutation from our population.
Just coming back to that, whether this mutation was earlier reported in Lahore?
No. Novel, as the title shows, that this is the novel mutation.
Because when you assign it a pathogenic, you did some functional assays on that.
As in the diagnosis, at the steps of diagnosis, we need to diagnose what is the cause of the disease. If there are reported variants which the function is not known or for the specific gene, then we will go for the functional analysis. This could be the second part of therapeutics, but not this way.
Quite straightforward because the variant is not earlier reported.
So these are, as we know that these are rare autosomal recessive disorders.
You assign it as a pathogenic.
Yes.
Likely pathogen is more relevant. Why going back?
Thank you so much. Our next speaker is Dr. Sana Khabar. She is from Dr. A.Q. Khan Institute of Biotechnology and Genetic Engineering, University of Karachi. She will present her research involving whole exome sequence.
Bismillahirrahmanirrahim. Assalamu alaikum. Good afternoon, everyone. I'm beyond excited to be presenting a bite-sized finding from my PhD dissertation. I'm a PhD pursuer at Dr. A.Q. Khan Institute of Biotechnology and Genetic Engineering, University of Karachi. And the title shows that it is whole exome sequencing study that identifies a novel variant which is associated with Angelman syndrome associated variant in autistic broadband. It is basically the finding from my broader title, the dissertation, that is genetic variations in GABRB3 gene and their association with autism spectrum disorder. The objective of the study, basically, we have got I'll be in a hurry, honestly, because of the lack of time. So we have got three broader objectives of the whole study, and the findings that I'll be presenting today belongs to the second objective, that is to identify a common genetic landscape of severely autistic patients with silent seizures and regression, and also the intellectual disability. So this boils down to the hypothesis that is the exploration of overlapping phenotypic features of ASD patients. They may imply a common genetic marker that could be associated with the severity and complexity of this condition. To introduce the autism, it is basically characterized as three basic characteristics. One is the communication and socialization impairment and very restrictive and restricted behaviors. And this whole situation actually becomes even more puzzling because it is associated with many genetic variants, which again make it can be syndromic ASD because of the copy number variants and the SNPs and all. And also there could be very behavioral challenges like SPD and ADHD, depressive disorders. Also the neurological challenges, seizures and microcephaly, and metabolic disorders like like GI disturbances and enzyme deficiencies. And in addition to that, there could be intellectual disability and language impairment. I would like to call it a real pandemic because of the prevalence of this disease, which is increasing every year. So this data is from this graph shows the data, the CDC report from 2018, which says that 1 out of 54 birds are affected with autism. And the recent report says that 1 out of 44 birds are affected with autism. And the report that is about to come out now is going to say that that 1 out of 36 birds in United States of America are affected by autism. And globally, 1 out of 100 birds, one child out of 100 birds is going to be affected by autism. So by 2050, we are going to have 1/3 of the population with autistic adults, which is alarming. And coming to the ASD-related syndromes, which are due to because of the known changes in some genes, which could be, which can lead to the Fragile X syndrome, Fragile X syndrome, Rett syndrome, Angelman syndrome, and Tourette syndrome. This particular study is the case of a child who was presented as an ASD, a 5-year-old female patient, but after the whole exome sequencing, it was found to be an Angelman syndrome and not ASD. So this is about Angelman syndrome. I will just quickly go through it. The prevalence is about 1 out of 15,000 births, and it's the known cause is the maternal UBE3A gene, which is located at chromosome 15, and that is also maternally imprinted. So the prognosis is that autonomy is never achieved. The patients usually they need to have a life support, and this is an AI-generated image when you put on all their their features, and the technique, the tool actually generated this image. And these kids, the Angelman syndrome patients, they are developmentally delayed, they have this strabismus in their eyes, small nasal bridge, and they have very distinct facial features, and they can be identified through, just by looking at them. But having said that, we do miss out the diagnosis. So this is the UBE3 gene, which is located at chromosome 15. Oh, I have this. This is the chromosome 15, and this is, we have got like 2 copies, the paternal and maternal, and this is basically only the maternal copy is imprinted in the neuronal cells, whereas the paternal copy, it stays silent. So whenever there is one SNP or one mutation or one loss of the information, if there is any deletion, the expression will become as an Angelman syndrome. So the methods involve the ethical approval from the IRB of the KIPG Institute, and the informed consent for publication for the participation was taken, and the patient's data, the detailed data was collected. The samples were sent to Centogene for the whole exome sequencing, and there was again the pipeline establishment and the data analysis. So this is the patient's history. There was a placenta previa reported during the pregnancy, and then there was APGAR score. There was regression during 1 to 3 years of her life, and then there was no speech, absolutely no speech, absolutely no eye contact, absolutely no social reciprocal smile. And there were sleep disorders, motor dysfunction, ataxic gait, and this is the parental profile, the non-consanguineous parents, and yeah. So we just moving on to the results, because this is the pipeline that we have been following for many sessions. So this is the results from the ensemble, which shows that this single patient, this single data set had 308,000 something variants that were processed through it, which had a lot of missense, synonymous and frame-shift mutations. The pathogenic variant that was identified was a novel variant, and there was also, and which was the primary finding, which actually caters to your question, that we actually matches the finding. The finding that matches to the phenotype the most is the finding that we identify as a rooting cause of any disease. IDUA and MTFHR was the secondary finding, was reported to be the secondary findings. Then the string analysis was performed to show if there was any pathway overlapping of the genes, and these identified genes did show some overlapping of the pathways through the KEC pathways, except the one which is the IGUA gene, which is reported in Pakistani population as a marker for intellectual disability in some publications from Lahore Children's Hospital. So quickly, I'll be summarizing it here, that this is the Q arm of chromosome 15, and this is the UB3A gene, which has this novel variant, and this is the GABA-B3 gene, the GABA receptors gene that I'm working on the broader spectrum. So the whole area becomes our focus, and and these are the genes here, the Prader-Willi syndrome and the Angelman syndrome, and these are the imprinting centers. So the whole area becomes our focus of attention for intellectual disabilities, seizures, and regression in this neurodevelopmental disorders. Then we predicted this protein structure, which this is the protein that was identified to be in the HET enzymatic domain. Sorry, sir. So, as to conclude it, the whole phenotypic neurodevelopmental disorders, since they have similar type of phenotypes, so there's a chance of missplit diagnosis. So whole exome sequencing and NGS is the only way to go forward. I would also like to propose some future prospects like the trios and the whole exome sequencing, and also the epigenetics do play a part. I would also like to acknowledge my team here. This is our medical wing of KIPG. These are the references. Thank you very much for your patience this time.
Thank you very much. Now I would like to call our last speaker, Dr. Syed Ali Raza Shah from Khan University Hospital. He will present his work.
Thank you, Assalamu alaikum. So I'm working as an assistant professor in the Department of Pathology and Lab Medicine. So my talk will be related to the study of association of mitochondrial variations with the severity of COVID-19. So the COVID-19 was all around in the globe as well as in Pakistan. So considering, and you can see the facts and figures you will be hearing around all the time in different speeches and talks. I'll not go into the details of those. So the background of this study to design a study to plan the association of mitochondrial genomics with the severity of COVID-19. The reason was that the mitochondria was at that time it was recently emerging that different viral diseases have the impact in context of severity with the mitochondrial variations. So we designed a project in which to complete mitochondrial genome sequencing and see that whether it has any impact, whether the variations which we got, both in terms of the haplogroups at the broader scale and the specific mutations at the smaller scale, the specific scale, have any associations with the severity of COVID-19. So I'm not going into that detail. So one thing that we have considered is that the mitochondrial haplotypes are also the markers of the distinct populations as well. That every population has a distinct mitochondrial haplogroup that can also trace back its history, and that also have some things in common as in perspective of being vulnerable to a particular disease or being resistant to some diseases. So some of the articles which have came out regarding the study of association of mitochondrial haplotypes with and the variations with the severity of COVID-19 were from India, Turkey, and some from the China, where they have identified that some mutations specifically in ND1 region and some non-coding regions were there, which are responsible for the severity of COVID-19. So considering that we have developed an objective, I've already highlighted that. So the methodology was, there were 4 groups in all, but right now I'll be talking about just 2 groups. The groups One group was asymptomatic mild, and we recruited 70 samples. In fact, we got the samples from the biorepository from the Afan University biorepository, and there were 54 samples who were severe or critical COVID-19 patients. And these are according to the WHO classifications, these are characterized according to the WHO classifications. We had the samples, we collected the genomic DNA and amplified the mitochondrial DNA, and did the sequencing and did the bioinformatics analysis. So these are the haplotypes, and as you can see that the M haplotype was one of the major find in context of the haplotypes, which was in case of the mild in case of the mild or asymptomatic mild group, the M haplotype was more prevalent as compared to the critical severe group. So this to some extent shows that the M haplotype is more sort of protected for the severity of COVID-19. Then we dive deep into the total variations. There were a total of 349 variations in the asymptomatic mild group. 114 of those were shared with the critical severe groups, those variations, and 463 were overall in AMD group, 178 variations were overall in the critical severe group. Out of those, 114 were common between CS and AMD, and 349 were distinct for the AMD group, and 178 were distinct for the 64 were distinct for the CS group. This chart shows that what are the mutations present in different mitochondrial coding region genes. This graph shows that what are the variations or the frequency of variations in the non-coding genes of mitochondria, that is also called the control region. And there were several sites where there were statistically significant associations also found. I'll be talking that in the next slide. Yeah. So after doing the Fisher exact test, we found that all those which are highlighted as red are a sort of red have the positive association with the severity of the COVID-19. And these are the details of those variations, and we've also put it into the online databases, the MitoMaster. So they help us to identify the different scores of it, and the higher scores we focused on, especially the mutPred score, because for the mitochondria this is one of the baseline to identify the pathogenicity of a particular variant present. And we identified that there were many mutations which were variations of the same, which were identified through the sequencing in the critical severe group. Many of those were not many, in fact, almost 10% of those have the mutPred score of more than 0.5, showing them that they are computationally they are pathogenic. And these are the side chains which have been changed because of the variations. And this is the snapshot of just two topmost variations, and the scores from the different databases, which that ultimately shows that they actually have the pathogenic part. So I'll conclude that we found some haplogroups. There is M which is more vulnerable to the severe COVID-19, and there are several distinct mutations which we have found through this study, and we'll be taking them together. We'll be doing more of the computational related stuff, and we will shortlist few of them and expand this study, because we have two more groups. There is the healthy controls and the moderate group of the COVID-19. So we have compiled the complete data of that one as well, and will make a whole the complete conclusion based on all the four groups. This project was funded by the HEC, and this is the ERC approval of the bill. USTRA did most of the sequencing related stuff, and these are all the contributors. Thank you so much.
Thank you so much. Thank you so much. We have come to the end of this session. I would like to invite Dr.
Thank you.
Okay, so Rana was the friend replacing her because she had to go to another session. So I'm Dr. Reena. So this is the last talk of the session. I would like to thank the scientific committee for putting up an interesting talks here. the session was emerging trends and technologies. So they really have kept the title, and we set the stage by Elena, Dr. Sami, Dr. Catherine, by nanotechnology to give us the emerging technologies. What do they have? And then Dr. Faisal has showed us how he used all these technologies in precision medicine. And all these free papers, we have gone from targeted panels to whole exome to whole genome, and really interesting results and papers. So I won't take a long time because we are ready to give, because you know that these oral papers are, our judges were judging their talks, keeping in mind the time, the presentation, the confidence, the question answer, and all those things. So hopefully I'll pass it on to Dr. Natasha, and thank you very much.
Thank you so much, Dr. Ramina, Dr. Sadat, for for chairing and moderating the session. Just to wind up before we take a short tea break, and then we need to come back here by 5 o'clock, please, to start session 2. We've got some very exciting talks, especially related to clinical research and translational research in Pakistan. But just to thank our participants from session 1, I would like to invite Dr. Said Khan to present some shields and certificates, please.
I would first like to invite the session chair, Dr. Rana Muzaffar.
I'm sorry, Dr. Rana Muzaffar has to go to another session. So.
Okay. Session co-chair, Dr. Ramina Kazi. Dr. Sadat Ali, the session moderator. Dr. Faisal, the invited speaker. Dr. Catherine Warren, but I guess she has left. And Dr. Ramina, I would like to invite you to give it to Dr. Said Khan, our invited speaker.
Define. Define.